Hi and hello everyone. In this very pleasant morning, I am Partha from the Biomics team. Would like to welcome you all for today's session. The speaker of today's talk is Dr. Vivek Dogra, a senior scientist in CSIR-Institute of Himalayan Bioresources Technology, Palampur, Himachal Pradesh in India. He will be delivering his talk on plant-environment interactions. What does chloroplast do in there? On behalf of the Biomics team, I would like to welcome our eminent resource person, Dr. Vivek Dogra. Our speaker is an eminent scholar in the area of plant molecular biology, cell signaling, functional genomics, and proteomics. Henceforth, I believe that he would be nourishing you all through his talk. At this juncture, I would like to thank Professor Dr. Vivek Dogra for accepting my invitation and taking the time out of his busy schedule to join with us. It's a highly honor for our Biomics team in hosting this webinar with an eminent resource person. And I also appreciate and welcome all the participants internationally across the world who joined with us for this talk. Dear participants, we have question and answer sessions at the end of this presentation. You are supposed to place your questions in the chat box. Myself here to put your questions to our guest speaker during question and answer sessions. And also you can place your feedback as comments in the same chat box of the YouTube. Before we start this session, it's my pleasure in taking a credit of introducing our speaker, Dr. Vivek Dogra. Dr. Vivek Dogra is a senior scientist in CSIR-Institute of Himalayan Bioresources Technology, Himachal Pradesh in India. He was a postdoctoral fellow in photosynthesis and stress signaling group in Center for Excellence in Molecular Plant Sciences at Sichuan Center for Plant Stress Biology, Sichuan in China. He received his postdoctorate in PhD Plant Biotechnology in CSIR-Institute of Himalayan Bioresources Technology in India. He did his master's in MSc Molecular Biology and Biochemistry in Guru Nanak Dev University in India. His research falls on plant molecular biology, cell signaling, functional genomics, and proteomics. He is currently interested to understand the role of the chloroplast for sensing and managing various environmental developmental cues and the cognate responses and trying to unveil the readjustments in the chloroplast homeostasis and to delineate various retrograde signaling cascades initiated from the chloroplast using the model plant Arabidopsis thaliana and high-altitude extremophiles of southern Himalayas. Since the changing climate poses a significant challenge as it presents distress to the growth and development of plants. Therefore, there is a need for plants to develop adaptation strategies. Ample evidence is in recent past, projected chloroplast as sensors for environmental clues, which priming the plant response to deal with the adversities. He is a recipient of several awards such as Ramalinga Swami Re-Entry Fellowship 2019 to 2020 by Department of Biotechnology, Government of India. President's International Fellowship Initiative, Postdoctoral Fellowship by the Chinese Academy of Sciences in December 2018 for 2 years. Dr. D.S. Kothari Postdoctoral Fellowship by University Grants Commission in Government of India. Qualified CSIR UGC NET lectureship in life sciences twice in June 2016 and December 2008. Qualified radiology safety officer level 1 examinations at BARC India in 2007. University gold medalist in post-graduation aspect. With this brief intro, may I now request Dr. Vivek Dogra to deliver a lecture on plant environment interactions. What does chloroplast do in there? Sir, please.
Thank you, Partha. So, I'll start first sharing my slides. So, is it visible now, or?
Yeah, you can go for slideshow.
Okay, perfect. Okay, so yeah, you can now. You can see my slides. Okay. So first of all, I would like to thank Partha and the whole team of Biomics, to really bring all of us together on this platform. And I would also like to thank COVID-19 particularly, because of the COVID-19, now it has it gave us a chance to develop such platforms where we do not even have to travel to meet different people. We have now virtual modes where we can organize meetings and seminars, discussions, and so it is quite economical and safe. And I would also like to thank everyone who is presenting here and who is watching right now, and also would like to thank all the people around. So I will start with my slide, and the title is Plant Environment Interactions, and what does chloroplast doing that. So since we talk about the plants, and we know the plants are organisms which can make their food by themselves, because of one organelle they have, that is called chloroplast. And because the chloroplast plants are special, they are different from us, like humans or other organisms. Okay, so in the past, actually, when the life was originated, it was not like that. We were having eukaryotic cells just like that, but they evolved by time. And mitochondria and the chloroplast, what we see in today's plant cell, so, and in the animal cell we see only mitochondria. So anyhow, these two organelles, they entered eukaryotic cells via endosymbiosis. So before that, they were like unicellular cells by having single genomes. But later on, these endosymbiotic events happened around 2.7 billion years ago. And what happened that time is a prokaryotic host was attacked, by an alpha protobacterium, and that actually entered the prokaryotic host cell and became a non-photosynthetic proto-eukaryotic cell, which resembles like human cells, or animal cells, having only the mitochondria and the nucleus. Now, this cell was having two genomes. One was from host in the nucleus, and another was in mitochondria, and that is from the intestine. So at the same time, there was another event happened, and this time a cyanobacterium, which is slightly evolved, kind of a photoautotropic cell, bacterium that could, then enter this non-photosynthetic proto-eukaryotic cell, and it now became a photosynthetic eukaryotic cell having three genomes, encompassed in three organelles: chloroplast, mitochondria, and the nucleus. Okay, so this actually happened during I think around 2.7 billion years ago, and it was kind of a continuous process and continuously evolving. So what happens during now this evolution? The endosymbionts, they transferred their genomes to host. So what now happened is, like plastid and the mitochondria, they transferred around 90% of their genomes to host genome and integrated them in the nucleus. Now, this was because these cells, the endosymbionts, they were not able to work independently, and they were having a lot of dependence upon the host. That's why and you all would be aware that maintaining the genome itself is a energy consuming process. So what these endosymbionts did smartly, that they transferred their genomes to the host so that they can have, they can relax, and they can like do other things for which they were brought toward in the host cell. So because of this, now what happens now, this chloroplast and mitochondria, they always get in touch with the nucleus, and they maintain the harmony via two kind of signalings, which we call anterograde and retrograde signalings. So anterograde is something which when comes from nucleus to the organelles, or is called forward signaling. And when something is coming back to nucleus, some signal is coming back to nucleus, we will say it is a retrograde signaling. So mitochondria and chloroplasts, they talk to nucleus via these two signalings, and at the same time they also talk to each other via some other signaling means, especially via the chemicals. Okay, so and as we all know, so now what happens during the metabolic reactions, we see a generation of a lot of reactive oxygen species. This is the beauty of the biochemistry or the photochemistry, where we see the transfer of electron is important, to make different molecules, and these molecules could really make the energy in terms of NADPH and ATPs. So for the generation of ATPs and NADPH, which happens in different organelles, especially in the chloroplasts and mitochondria, so they actually, during this process, they generate a lot of reactive oxygen species, because oxygen has a chemistry, such a chemistry that it can absorb light, electron. It can accept electrons because of its chemical nature, and at the same time, its chemistry also allows it to gain energy. So because of these two processes, by gaining energy and the electrons, the ROS are formed, and these ROS includes singlet oxygen, which actually specifically generates in the chloroplast, and superoxide anion, hydrogen peroxide, hydroxyl radicals, and many nitric oxide species also gets generated in other subcellular organelles, including mitochondria, peroxisome, and in the plasma membrane, like in lipoplast per se. So in the when we talk about the chloroplast, so we can also see the similar thing happening in the chloroplast, mitochondria as well. But if we see the chloroplast, it has more kind of ROS than others. Why? Because in the photosystems, when the photosynthetic electron transport chain runs, there are 4 different kind of ROS which gets formed in the chloroplast, and all other organelles will have 3. The difference is the singlet oxygen. The singlet oxygen is one of the ROS which is generated upon transfer of energy to the oxygen. So what happens, we all know, when the light incidence on the chlorophyll, and the chlorophyll, because it's a photosensitizer, it's a pigment which can absorb light, so it absorbs light and it becomes excited. So now chlorophyll is stored in the photosystem 2 protein. It is bound to the photosystem 2 reaction center proteins. Now once it absorbs energy, the photosystem 2 becomes highly oxidant, and it releases 1 electron to the plastoquinone and the pool and towards the photosystem 1. And during this process, now photosystem 2 has to come back to this ground state, but because it's an oxidant, it is highly energized. It breaks the water molecules to gain its electron. At the same time, it comes back to the ground state by releasing its energy, and this energy is gained by oxygen, and this oxygen becomes oxygen. And then in the photosystem 1, we will see the generation of superoxide anion, and then which is catalyzed to hydrogen peroxide, which is less toxic comparatively, and then that is further scavenged into non-toxic water molecule. And during this process, hydrogen peroxide, hydroxyl ion can also be formed. So this happens normally all the time in the cell. But what happens when we have stress or developmental interventions? It's like during development now, for example, seed germination is a developmental process, or photomorphogenesis is a developmental process, or seed flowering is a developmental process. So that time there is kind of acceleration in the this metabolic reactions, and that will lead to gain of this. Lead to accumulation of this ROSs, which have some certain functions in the development. At the same time, when the plant faces environmental challenges, including drought, high light, temperature fluctuations, salt and metal, and even the pathogens, although we see pathogens attack from the outside, but they can also, have an effect on the chloroplast itself. So upon these challenges, the ROSs starts, accumulating a lot. So when these ROS accumulates, so what happens now? So upon this, the plant, the chloroplast, actually, what they do, they either face oxidative damage because of the ROS, and then if the damage is bearable, then this ROSs can also be used to utilize to trigger retrograde signaling back to nucleus and to save their souls. Now chloroplast will talk to nucleus, say, okay, save us. and we are in the danger, because most of the detoxification proteins which will be working in the chloroplast are encoded by the nucleus because of the endosymbiosis and horizontal gene transfer. So now there are certain examples where we see the ROSs are the increased levels of ROS are implicated with instigation of retrograde signaling cascades, and there are many pathways and molecules which have been identified by different workers in the world, and in that we see a lot of molecules which are generated in the chloroplast, including like chlorophyll biosynthetic intermediates, or MEP pathway, like this metabolites. These metabolites can accumulate because of the disturbances by the ROS, and then these accumulated metabolites can instigate certain signalings via certain molecules to talk to nucleus, where the nucleus will change its gene expression, and then the further action will be taken to really save the plant, or let it go towards the cell death. So among these, we'll be talking about one of the pathways which is implicated with the ROS, and especially with singlet oxygen. And so for this, as I told in the last slide, the singlet oxygen is largely generated in the photosystem 2 complex in the chloroplast. And this is generated by the gain of energy. And I think this is something which is very basic, and this is happens at the molecular orbitals level, when in the atom, the molecular orbitals of the oxygen, in the last, the outermost orbitals, they have, according to Hund's rule, they have single electrons filled in their orbitals. But because the gain of energy, the spins of the electrons changes. Now opposite spins repel each other, and then this molecule becomes reactive. So that's how single oxygen is generated in the chloroplast. So as I told you, when this ROS will accumulate, it will primarily damage the molecules in the vicinity, and the molecules involves photosystem 2 proteins. So single oxygen is generated in the photosystem 2, close to the photosystem 2, and it actually targets photosystem 2 proteins itself. And because of this, the photosystem 2 proteins, which has the function to transfer electrons to the photosystem 1, they are attacked by this ROS and they become inactive. This process is called, photosystem 2 damage. And this photosystem damage then undergoes a repair process, where this damaged protein is degraded, and a newly formed protein is inserted into this complex, and then this complex again go back to the active form, and then it starts working again. So this is a photosystem 2 damage repair cycle, and it is an inevitable process which always happens in the plant, in the chloroplast, in any condition. So what happens is, like when the plant is under stress, the rate increases, and when the plant is under normal conditions, the rate is declined, but still it always happens. And here FTSH proteases, which are the homologs of the FTSH proteases which are found in the bacteria, they still are the same, and they have a similar function here as well. They make a heterocomplexes here. So based on the levels of this single oxygen, they can have a different fates. So if under normal condition the single oxygen is accumulated, it will be quenched, and the plant will have no problem, and it will grow normally. But however, when the levels increases, so they could be look like sublethal levels, where the plant still can survive, it can still bear the ROS levels. In that condition, the plant can activate two kind of signalings. One can lead to the acclimation, where the plant growth will slow down and it will sustain. Or the plant can also decide to kill some of its own cells by activating a programmed cell death. So to sacrifice some of the cells which are affected more, to save others. But if this level of ROS goes much beyond this level, then the plant has no option, to return back, and the plant will, ultimately die. So depending upon the levels, the plant can have a different responses based on the signalings activated by the levels of ROS per se. Okay, so as I told you, single oxygen can activate signaling cascades. So So there are like 4 set of signaling cascades which have been, characterized until now. So out of this, as we know, ROS can target the biomolecules, and these biomolecules includes what? These biomolecules includes proteins, lipids, DNA, and also the carotenoids. Carotenoids are the molecules which we also think about like antioxidants. So carotenoids has a function, to scavenge these ROSs. So Singlet oxygen is scavenged by one of the carotenoids, which is called beta carotene, and there are other xanthophylls and lutein and few other carotenoids. They also have the similar function. Even tocopherol, vitamin E, it has a similar function to use to scavenge singlet oxygen or singlet oxygen derived molecules. Now, when the singlet oxygen or the ROS, this ROS is targeting these biomolecules. These different biomolecules will be affected at different levels of the ROS, and they will instigate different set of signaling pathways, which will be leading to different responses, including acclimation or cell death, or maybe detoxification, or sometimes there will be immune responses as well. So in this, so when I in my postdoc, in my work, I mainly worked on establishing these pathways, and I worked on 3 of these pathways, and I will be presenting I'm starting with 1, and if we have time, we can go further. So this was the first thing which actually was devised to show that ROSs can also instigate signalings, and they can be signaling molecules as well, besides being toxic. So the story began 20 years before, in 2001, when Professor Klaus Appel's lab, they found one mutant in Arabidopsis. So normally, Professor Klaus Appel was in ETH Zurich, and he was working for like 30-40 years, around 1965 he started his career, and until 2000 he was still working with the chlorophyll biosynthesis. During his work, what he found that in the chlorophyll biosynthesis, the chlorophyll biosynthetic intermediates, they all have the pyrrole ring, and they are called tetrapyroles. So all these tetrapyroles has alternate double bonds, and they have a tendency to harvest light energy. So they are called photosensitizers. They can absorb light. Once they absorb light, they become excited. So this is not good. So once they get excited, they will transfer the energy to the oxygen, and they will make a lot of ROS. So therefore, there is a regulation in the biosynthetic pathway. So what happens? So in the chlorophyll biosynthesis pathway, the last step of this pathway is catalyzed by an enzyme which is light dependent, which cannot function in the dark. So the system has to be switched off when we have dark, so that the chlorophyll intermediates do not get formed and do not get accumulated. Otherwise, what will happen as soon as the light will be switched on in the morning, or the sun will rise in the morning, the plants will have a lot of this tetrapyroles accumulated, and they will all start absorbing light. So some of the part of this accumulated tetrapyroles will make chlorophyll, but most of them will make a lot of ROS. That is not good for the plant. So that's why what plant did made a system to switch off the first enzyme of this reaction by a protein, and this protein was named as flu because of its phenotype. And now when this protein, so now this is one example. The wild type plant, if it is grow in the dark, then when we switch on the light, it grows normally. However, if this protein, the flu protein, has a problem, it loses its function, then what happens? That there is no switch off now in the night, the dark, and the tetrapyrrole starts accumulating in the dark, and as soon the light comes, they generate a lot of ROS, and that ROS is not good for the plant, and that kills the plant. However, if this dark to light transition is controlled, then the plant can survive, but it shows growth inhibition phenotype. Okay, so this was interesting to see. Okay, then before they thought ROS can always kill the plant, it always impose damage, but it was also found that it can also have a signaling aspect, and then plant can still survive. It means there is a system to deal with this with the ROS. So to really check this, whether this is genetically controlled, if this is genetically controlled, because the problem is in the chloroplast, it should have talked to the nucleus, to save it. So they did microarray analysis, and they found when they give dark to light transitions in the plant, in the flu plant particularly, they see a lot of gene expression changes, which include some of the genes which can activate the detoxification process and save the plant. So this was interesting to see ROS can activate signaling, and that signaling is genetically controlled. Okay, so this was very kind of changing discovery where people had changed their view towards the ROS, that ROS can also be can be a signaling component. So what now to find how this signaling happens? So what Klaus Appel's team they did, they induced random mutations in this blue mutant using chemical mutagenesis. They used EMS, ethyl methylsulfonate, and they found 16 plants, which could survive this dark to light transition, and they were having no problem. The plant was happy, plant was able to survive like wild type. So even they were not having this switch off, something was missing, and these plants were not able to tell nucleus, and the plant was happy. So this was very interesting to see, and then they mapped the mutation, where this mutation happened, and interestingly they found 16 such revertants, and all those 16s were having mutations in a single genetic loci in one of the genes per se. So in this gene, 13 were nonsense mutations, mean they were making stop codons. So because of the random mutation, a stop codon came into the open reading frame of the gene, but out of those 3 were the missense. They were making some other amino acid, but because even of the different amino acid, this protein was not able to function. So now this was named as Executor, because the presence of this gene was executing the plant, and then when this gene was knocked out, this gene was was losing its function, the plant was surviving. So this gene was named as Executor 1. So this was again compared when this gene was again put back into the this double mutant background, and this again behaved like flu. So it was confirmed that EX1 is the gene via which or the protein by which the signaling is being transferred or translated. So then what they did gene expression analysis also, and they found all of the genes which were upregulated in the flu upon dark to light transitions, they were repressed in the EX1 flu, and there was no signaling, and plant was happy. So EX1 flu plant was just behaving like wild type plant, having no problem, despite of the fact that it was having a problem in the chloroplast because of accumulation of tetrapyrrose and singlet oxygen, ROS. Okay, so the question now comes how EX1, this Executor 1, we named it EX1 also. So EX1 mediates oxygen signaling. So how this protein can work in the signaling. So this was when I started working with this protein, and what we found that this protein is actually localized in the chloroplast and in the thylakoids, and it's a membrane protein, and it is localized in the chloroplast per se, and this protein is localized in the thylakoids also. We can further subtractionate its localization, and we found that this protein was localized in the outer region of the grana stack, and this region we called grana margin. So margin region of the grana, this protein was localized. And if we see the protein structure, protein has a single peptide to be targeted to the chloroplast, and this protein has a UPRBC domain and domain of unknown function, DUF 3506, and this was at the C-terminal end. So this protein was until now having no function, not characterized. So we started working with this protein. So what we found that this protein, when we give dark fluid transition or single oxygen dose, we see this protein undergoes a degradation, whereas the control proteins were remaining similar with stress. So this was very interesting phenotype to see this protein undergoes a degradation process during sensing the single oxygen. So something is happening to this protein that is making this protein to undergo a degradation. Okay, so to check how it happens, so what we did pull down assay. What we did, we tried to take out if this protein is getting degraded, there should be some enzyme which should be degrading this protein. So what we did, we tried to pull down its interactors and its associated complex. Means the proteins with which this protein is interacting. We try to collect all of them and identify using LCMS approach, and we could identify a lot of proteins which were making complex with this Executor 1. So interestingly, this Executor 1 protein, as expected, was interacting with the grana margin localized proteins, including photosystem proteins, which undergoes repair process at the grana margin region, and chlorophyll biosynthetic pathway genes. And at the same time, it was also interacting with FTSH protease. So FTSH protease is composed of 4 subunits: FTS 1, 2, 5, and 8. And this EX1 was interacting with all of these proteins. So this was very interesting. Now, what we thought, if FTSH was the protease with which this the protein is undergoing degradation, so what we did, we made a double mutant in the flu background by crossing 2 mutants, and we created a double mutant. And what we see in the double mutant, the protein EX1 was stabilized. There was no degradation. So it was having a problem. It was sensing the single oxygen, but it was not undergoing degradation. Okay, so it was something which we saw very interestingly. Something is happening to the protein per se. So protein is having some problem. It is sensing the ROS, but it is not undergoing degradation. Okay, then what does it mean? So if we are correct, we thought probably there will be no signaling and there will be no cell death. Okay, so exactly this was happening. So when we gave the dark fluid transition to this FTSH2 and flu double mutant, so this now flu mutant has no FTSH protease, so it will be not having any degradation of Executor 1. So there was no cell death. The plant was able to survive, and the flu mutant alone was able to die. It was dying, and the flu mutant was able to survive. And it was not only at the level of the plant per se, but at the subcellular level also, we can see there was no cell death. The cell was having no problem. And the gene expression was also not changed. All the gene expression changes were reversed in the FTSH2 background. So flu FTSH2 double mutant behaved just like flu EX1. It means we found another downstream component that ROS is sensed by EX1, and EX1 is then when EX1 senses ROS, it is cleaved by the FTSH protease, and then it makes something which is leading to the signaling. Okay, so the question became how EX1 becomes the substrate of FTSH protease. So what we now went back to the protein structure, and as I told you, protein structure has 2 domains, UPRBC and UF3506. So in the literature, UPRBC is known to have function in the protein interactions, whereas domain of unknown function 3506 was having no function at all. So until now, there was no function reported for this. And we also did some phylogenetic analysis and other analysis, which are not showing here. So this is exclusively found only in this protein, not in any of the other proteins in the plant cell. This was something very interesting. So then what we did, we just took out this domain from this protein. We took out this protein domain, and we made an Executor 1 protein overexpression line in the EX1 flow background by deleting these 2 domains. Now we have 2 systems where we had EX1 whole, and in one system we just deleted this domain. So what we found, the E-exon protein was undergoing degradation upon single oxygen dose, but when we deleted this DOF domain, there was no degradation. Means this domain is important to sense ROS. So this was later we named our ROS sensor. Anyhow, so not only in terms of the protein per se, but plant per se was also having no problem, similar result. So when we delete the domain, it was also behaving just like FTSH flu and E-exon flu. So by deleting the domain, we can inactivate the function of the protein, and also can say that this domain is important for sensing the ROS. Similar thing was happening with the gene expression. Okay, this was interesting. So how this happens? Then again, we know if EX1 is sensing the ROS, means it should undergo oxidation by itself. So we now then try to find the oxidations in the EX1 protein. So we did the same co-IP immunoprecipitation assay, and we use mass spectrometry and then post-translation modification analysis. And interestingly, what we found, and this was again very interesting phenomenon, that we found oxidation in a tryptophan residue in this domain. So interestingly, this was something very new for us, that tryptophan is undergoing oxidation, because until now we thought, as a biologist, I always studied about cysteine as one of the residues which undergoes oxidation and make disulfide leakages. But this was something very new. Then we go back to the literature, and then what we found, tryptophan is the amino acid which is most prone and most susceptible for the single oxygen, and it acts as both chemical and physical scavenger of single oxygen in the protein per se. So this was not wrong, but kind of right, and we actually reproduced something which actually people discovered like 30, 40 years before. So this was very, very interesting. And we found that upon oxidation, the tryptophan, this is the tryptophan residue, and this tryptophan can undergo single oxidation or maybe double oxidation and make different variants, including NFK, kynurenine, and YA. So these all were detected in our mass spec data, and we could really see whether these were accumulating upon oxidation, upon sensing the single oxygen. So what we did next, we tried to mutate this tryptophan. We changed this tryptophan with the singlet oxygen insensitive amino acids, that is leucine and alanine. So alanine and leucine, they do not respond to the ROS because they do not have double bonds in their structure, and they remain inactive, insensitive to the ROS. When we changed this tryptophan with this leucine and alanine, the plant was having no problem again. So it means by changing one amino acid, we again changed the function of the protein, and it was behaving just like by deleting the whole domain. Either we delete whole domain or we just change one amino acid, we see the same phenotype, the protein become insensitive to the ROS. And similar thing was happening with the gene expression. The plant was happy. So this was actually in the DOF domains. So we named this DOF domain now as singlet oxygen sensor domain. So now we named it as a SOS domain. This is something we did last year. And so we made then a model that upon sensing the light, plant senses light, and then it makes the tetraphluols to photosensitize, and they make ROS single oxygen, which is sensed by the executor one protein, and then this undergoes a degradation by a protease and makes something. Actually, until now, we do not know what this protein makes, because until now, there is one hypothesis that something like the degraded products can come out to the nucleus, but until now, there is no report that a protein can come out from the chloroplast. There is always reports about the import, but there is no report about any export of a protein. On the edge of the plant. If we have time, I can share few more slides. Partha, we have time?
Yeah, we have time.
Okay, so then we also then while working on this this pathway, we actually found a novel pathway then last year. Actually, this was published this year only. So this was my actually second postdoc project. So what we did is we were trying to work on the EX1 mediated signaling only, and we were trying to find the downstream components. As I said, we do not know what are the downstream components. So actually, my bosses previous lab, they were working with this lab and this project, and they were trying to find out the downstream components. For that, what they did, they again used this power genetics approach, and they took one Singlet oxygen responding gene as a marker, and then they fused that with the luciferase. They use the promoter of that gene. So it means this promoter will sense the ROS, and then this will be activated. And then what we did, they fused luciferase as a reporter gene with this gene. So now what will happen whenever there will be ROS, this promoter will express the luciferase. So whenever when they give the dark to light transition, there will be ROS accumulation, and the plant used to flourish, luminous. So during this, they found many different mutants. Out of those, interestingly, what they found one mutant which was having constitutive activation of this gene. Means whether it was light or dark, even in the light, they were flourishing. Means something happened to these plants, and which were having this per se. Perhaps they would have accumulated more ROS, and that was leading to the activation of this gene, and this was independent of the dark to light transition, and perhaps this was independent of executor, probably, but it was something very new. So what it was done, that one of the mutant was CA33, and it was mapped, and it was found there was a mutation in one of the gene, and it's in its transmembrane domain, which is found in the outer envelope of the chloroplast. So then we actually started working on that, and then we found that this CRL mutant is having multiple lesions, multiple problems. So those problems included a cell cycle defect. Then this protein, loss of this protein was making chloroplast bigger. So see, this is one example of the wild type cell. In a wild type chloroplast, you see small chloroplast, and there are 80 to 100 chloroplasts in one cell. But in the CRL cell, the chloroplasts were not able to divide, and they were becoming bigger, and the number was reducing. At the same time, this plant, because of the big chloroplast, was also showing an age and light dependent cell death. So like 4-day plant was not showing any death, but upon 5-day cell death was starting, and this was increasing with the time. So this was something happening without any elicitation, means without any stress, the cell death was appearing by itself. So there is something happening, something is problem with the genetic program. So we thought we should use this mutant now to study chloroplast triggered programmed cell death. So we started this project like that. Then what we did, first of all we did RNA-seq. Now these days transcriptome analysis is kind of a popular technique. So we try to find out the genes which were changing upon onset of the cell death, and then we compared this 4-day and 5 days, and we did the RNA-seq, and what we found, there were a lot of genes upregulating in 4 and 5 days as compared to the wild type. So many of those genes were responding to the stress stimuli, and we named them as stress-related genes. And what we did, we then tried to find out what kind of transcription factors these genes are being dragged by. So we did this kind of promoter analysis of these genes, and we found 2 transcription factor families, MIX and TGAs. They were showing significant enrichment, and significant number of genes were being driven by these transcription factors. So then we looked into the details, what kind of genes these transcription factors basically derive. So what we found, these actually have some relation with the chloroplast, and the relation is that this mix and TGAs they respond to the jasmonic acid, like mix, they respond to jasmonic acid, and the TGAs respond to the reactive electrophile species. They are the molecules which are formed by the oxidation of carotenoids and lipids. So these transcription factors respond to these molecules. So what we see, ROS is in the chloroplast, they oxidize certain molecules, and then these molecules do lead to the activation of this transcription factor to activate downstream genes. This was something very interesting. So what we thought, okay, because in the plants, the salicylic acid and jasmonic acid are the known regulators of the cell death. So we thought probably they could be also have some function. So we tried to see how many genes are there which could be responding to SA and JA. So there were a lot of genes. And then for that, now to see whether they really have a function, we did again a forward genetics approach. What we did, we crossed the CRL mutant with the SA deficient and the AOS deficient mutants. Means these mutants were not having SA or JA, and we crossed them with the CRL. Now we have double mutants. We were not having the JA or SA, but both of them, like this is SA deficient mutant and this is JA deficient mutant, but still they were having cell death similar to CRL. So from here we thought, okay, JA and SA are not playing any role. So then what it could be? So then we thought probably ROS activated, RS oxidized products could be the molecules. If it is true, then we should see, then we thought we should be having a lot of ROS in this mutant. So what we first try to see is the accumulation of ROS. So for superoxide anion and H2O2, we have simple spectrophotometric analysis, but for the for the single oxygen, we do not have such thing, but we have a fluorescent dye which we can stain the ROS with, and we use that dye. This is called Singlet oxygen sensitive green. So it shows a green color if there is ROS. So what we found, yes, indeed, the CRL plant without any elicitation, without any stress, was having a lot of ROS perhaps because of its disturbed chloroplast structure. And we found there was a lipid peroxidation as well. So in the wild type, we see there was almost no lipid peroxidation in the normal condition. However, in the CRL, without any stress, there was a lipid peroxidation. And because of this lipid peroxidation, there would be cell death. But we also thought maybe it is because of the EX1. So we also checked whether EX was involved or not. So we crossed with the EX1 mutant, and we now knocked out the EX1 and actually, surprisingly, what we found, this cell death was still prevailing in the CRL-EX1 mutant, and this suggested us that the CRL, the cell death in the CRL was independent of the EX1, and the signaling was again independent of the EX1. So there was no role of the EX1. Then it means something new was there, and there could be a novel thing. So, and from here, we actually found that perhaps lipid peroxidation has a role. So means lipid peroxidation product could be the molecule which is instigating the signaling. So for that, then we also in the beginning started the forward genetics approach, where we did the EMS treatment to the CRL, and we screened many mutants. So many repressors. So one of the suppressors what we found was, we named it suppressor of CRL4. This plant was slightly bigger than the CRL, and it was actually interestingly had the same problem like CRL, had the big chloroplast, but surprisingly it was not having any cell death. So this was something very interesting, that when we found a mutant which was having no cell death, but it was still having some other phenotypes. Then we mapped this gene, and we found this gene had a problem. Mutant has a problem in a gene which is named as FAD5. So FAD5 actually was responsible. So what we did, we again use another allele and also complemented this, and we found indeed FAD5 was the gene which was having a problem, and because of this loss of function of this FAD5, the CRL was having repressed cell death. So now, as we know, ROS can actually target these molecules, and lipids are the molecules which are routinely targeted by the chloroplast. But the ROS, it was one of the things which we thought, okay, we have a hope that probably lipid could be the now substrate. So and we found FAD5. So FAD5 actually has a function to introduce the first unsaturation bond in the 16 carbon fatty acids. So 16 carbon fatty acids are the prokaryotic fatty acids in the chloroplast. So in the plant per se. So they actually are only present in the chloroplast and in the mitochondria, and they are present in the bacteria. And otherwise, the plant cell have 18 carbon fatty acids as the most abundant fatty acids. But this was something which was only found in the chloroplast, and this was all coming into the same. Now everything was coming into the same picture. Now we have ROS, we have lipids, and now lipids. In these lipids, we have 16 carbon fatty acids, which are only in the chloroplast, and the FAD5 would lead to the generation of this unsaturated fatty acids, and unsaturated fatty acids are the targets of oxygen. So if we now switch off this FAD5, we do not see any unsaturated fatty acids, and we do not have any target for the ROS, so we do not see any cell death. This was very good. And we actually again checked all the things at biochemical levels also and genetic levels also. All the things were coming to the same hypothesis, and results were coming into the same picture. So then we thought, this is only specific with the CRL, or it is a general phenomenon. So we actually again checked with some other chloroplast division mutants. So they were also having a similar phenotypes. They were having bigger chloroplast, smaller stature, and they were showing the cell death. But when we inactivate FAD5 in all of these mutants, we see cell death was repressed. So by this, we found a pathway actually, which actually exists in the system, and when the ROSs are there, they can lead to the cell death. So this is the model where we actually worked out to show that plastic division actually happens in non-stress conditions and lead to the cell division. Because the cell division, in the cell cycle, cell division, before the S phase, the plastid and mitochondria, they already undergoes division. So this actually is a prerequisite. The cell will not divide if the plastid or chloroplast will not divide. So under the stress conditions, we thought plastid division is affected because of the disturbance in the chloroplast, and then this lead to the cell cycle arrest and activates the cell death via lipid oxidation. This is the whole story, and so we also worked on another thing, but maybe some other time we can talk about this. And there were many other findings. We found some applied things also, but I go straight to the take-home message that plants, although are society, they have evolved various systems to perceive and respond to the series of environmental challenges. And this is very important that cellular organelles, especially endosymbions, mitochondria and the chloroplast, they have essential functions in determining these plant responses because they can sense the stress. The increased generation of the reactive oxygen species, ROS, we all know, they mark the stress, and they also initiate various signaling pathways to trigger various responses to deal with the adversities. And now you all will have a question that what would we gain from this? This is a fundamental science. So this fundamental knowledge can be utilized to screen, and or to develop or to engineer stress resilient plants. So we know what is the gene per se which can be targeted. So now we can use novel techniques like CRISPR. CRISPR is the hottest technique at the moment which we can use to engineer the plant. But because this is something which is like engineering, but we can also screen natural variations. We can have thousands of populations of a single trait, and then we can compare those things for a single trait, and we can screen a better, robust variety of a particular crop or a plant. So with this, I would like to acknowledge my fellow workers and my boss, Professor Chan Hong Kim. And then also I got some fundings there. And then recently I came back to India and I joined CSIR-IHBT Palampur. And I would like to thank Director CSIR-IHBT also to give me this opportunity. And I would also like to thank my funders here now. I have CSIR fundings, IHBT funding, and I also was recently awarded the Ramalingasamy Fellowship, and I will also acknowledge them. And I have recently started my lab, so I have 4 students until now, and I would like to welcome other people. if they want to join and or to collaborate, they are welcome, to CSIR-IHBT. This is the picture of CSIR-IHBT in the labs of Himalaya. Basically, this is Dauladhar range in the Himalaya. With this, I would like to thank everyone and thanks for listening.
Yeah, thank you so much, sir. Your presentation is quite interesting and so informative. It proves that your knowledge fascinates in the field of life science, plant sciences, and how plant interact and how chloroplast makes its roles for interacting with environment. I hope you have enriched the scientific temper in overlaying aspirants who are about to take up their research, and many who are carrying out their research in their respective domains. And sir, we had a huge number of participants throughout your talk, and we have some questions from Kim. Now I will forward one by one to you.
Sure.
The first question is that under one electron transport system, whether same modifications are possible for singlet oxygen?
Come again, please. What is the question? I could not.
Yeah. Under one electron transport system, whether same modifications are possible for singlet oxygen?
Okay. I don't know if I understood correctly. The question is like with the electron transfer, the single oxygen can be generated. Okay. So by gaining electron, we have superoxide anion, and by gaining energy, we get singlet oxygen. So singlet oxygen is not gaining an electron, but is only changing the spin. The outermost orbitals of the oxygen molecule, they change their spin. The electron should be, by the Hund rules, they should be in the same spin. But what happens with the stress, with the gain of energy, the electron changes its spin. When the changes of the spin happens, so it can also transfer here, and they start repelling each other. So this became in an reactive molecules. This now molecule want to come back to its ground state.
Yeah. The next question is that whether SA as jasmonic acid triggered the protein modifications, whether
Okay.
SA or jasmonic acid.
Yes. Until now, it's not clear whether they themselves can make the protein modifications. But indeed, yes. In case of SA actually is synthesized in the chloroplast. The 90% of the SA is synthesized in the chloroplast, 10% in the cytosol. So 90% of the chloroplast, when it is in the chloroplast, it comes out into the cytosol, and in the cytosol, it interacts with NPR1 protein. NPR1 is an oligomeric protein, which makes a oligomeric complex. But once SA comes and binds to this protein, SA is a kind of ligand now, a small molecule. It binds with the protein. Protein is a huge molecule. Once SA molecules comes and binds with NPR1 oligomers, they becomes monomers. So they undergo modifications and becomes monomer. Now they lose their interactions with other subunits. Now NPR1 monomer, it can go into the nucleus, become a transcription factor, and derive the expression of SA responsive genes, mainly leading to the cell death. Similarly, JA also interacts with the MYC2 transcription factors. But in that case, this is something else which just happens. But yeah, indeed, these molecules, because they are small molecules, they are elicitors, they actually can change the protein configuration, they can change protein complexes, and they can then lead to the function of the protein, or maybe they can inactivate the function.
Yeah. The next question is that what happens to the electrons which are excited from the atomic nucleus? Does electron acceptor plays any role, and how it exactly relates redox potential scale?
Yes. So see, like when we see a lot of photo inhibition per se, so the photosynthetic electron transport chain cannot work because the plastoquinone pool is highly reduced. So under the redox, when the redox changes towards the reduced state, the class 2 phenol, which is the acceptor of the electrons in the photosynthetic electron transport chain, if they are already reduced, they cannot accept electrons from the photosystem 2. So now what happens is now photosystem 2 cannot transfer electron, and then but it still can absorb energy from the light. Like chlorophyll in the photosystem 2 will absorb. It will be continuously absorbing energy from the light, but the photosystem 2 cannot transfer the energy to run the photochemistry. So means the energy is gained but is not utilized. Then this energy which is not being utilized will be making more ROS. So this will lead to the change again. This will lead to the change in the redox, and it will lead to the altered homeostasis in the chloroplast.
Yeah. The next question is that how energy production and carbohydrates metabolism is altered by alternation or mutations in the executor regions.
Again.
Yeah. Is how energy production and carbohydrate metabolisms is altered by the alternation or mutations in the executor regions.
Okay, it's a really great question. So until now we haven't looked into that, but what we try to see about the photochemistry. So photochemistry actually was not having any problem, but indeed this is something which we are actually working on. Because when we still have the ROS, but we do not have the signaling. So but still plant is happy. So how this is happening? This is something is a worth working question which we are trying to find out.
The next question is that one electron or two releases when water divides to pass PS2.
Again.
When one electron or two electrons releases when water divides to pass PS2 system.
Oh yeah, so actually water splitting happens after when the photosystem 2 transfer electrons to the photosystem 1. Then now this oxidant photosystem 2 is a highly oxidant because it has lost its electron. So now it's also having energy. So now this energized oxidant photosystem 2 breaks water to get its electron. And yes, indeed, it's like two electrons will be gained by one water splitting.
Yeah. The next question is that how CRL exhibits upregulation of stress-related genes.
Oh, yeah, this is a great question. Actually, so what happens is because of the mutation in the CRLs cannot function any longer. So now, because of that, plant is having bigger chloroplasts. Okay, so bigger chloroplasts are because they cannot divide. Now, as you see in the cell cycle, we see there is a G1 phase and an S phase. So actually, DNA is dividing, everything is dividing, it's replicating. But if we do not have cytokinesis, then what we see endoreplication or endoreduplication. So what we see, the cell size becomes bigger, like we see in the epidermis cells or ameboidal cells. They are trichomes. They have enlarged cells. The cell doesn't undergo division, but they undergo rounds of replications of the material, genetic material as well as proteins, but they do not divide. So they are, because of this, they get bigger. So it's called endoreduplication. So now chloroplast also and mitochondria also divide in the same fashion. They also have this kind of small cell cycle, the chloroplast cycle we say. So they will also have the similar replications of the things. But now because of the absence of CRL, perhaps it has a function in the the plastid division. So now plastid is not able to divide, or chloroplast is not able to divide, so it becomes bigger. Because it's become bigger, but it should have undergone division. So now bigger chloroplast has bigger challenges to manage. So it has like problem is in the thylakoid assembly. So, and if we go at the ultrastructure level, we see problems in the thylakoid assembly, and because of that, there is a problem in the assembly of the photosystems and light harvesting complexes. So, because of this stoichiometric difference, we see a lot of ROS generation. That leads to now this signaling for the regulation of gene expression.
Yeah. The next question is that what is the effect on plant when applications of any herbicide and mutating FTSH?
Yes. So this is excellent question. Actually, I was not having time, so that's why I couldn't show. So for the FTSH protease, if it has a mutation, if we inactivate FTSH protease, we see severe phenotype. We see plastic division defects, and we see white patches on the leaf. So it's called variegation. So we see on the leaf at certain places there is no chloroplast. So there is a plastic biogenesis defect. So chloroplast cannot really evolve. And because the herbicides particularly, so herbicides actually, although when we go beyond certain range, they also show the similar phenotypes, but at lower levels, they actually also have a problem, to deal with this ROS accumulation. So, I mean, we see ROS accumulation in some of the herbicides.
Yeah. The next question is that can CRISPR target chloroplast?
Yes, indeed, it's an excellent question. And actually, I have this my DPT Ramalingasamy project was on the similar thing. So I actually am trying to now edit chloroplast genome. So until now, CRISPR is not that successful. I think almost not successful until now. But one approach is homologous recombination. So through which we are now trying to do this. And I mean, pinpointing one single gene is difficult because you cannot, because one cell has 80 to 100 chloroplasts. And then to guess homozygosity and also even to target chloroplast genome is not so easy. But people are now doing with the root apical meristem, and there they are trying to with the stem cells of the roots, they are trying to develop the callus and doing gene gun based transformations there. So if somebody is interested, maybe they can discuss with me. Actually, I'm working on that. It will be nice to, talk.
The next question is that is the role of FAD is the same in case of photosynthesis and respiration?
Okay, I will not comment on that directly because I do not really have an answer for that. But perhaps they have, although because they are the biomolecules, they have similar functions, but in different pathways, they can have a different impact. So although their chemical or biochemical function will be the same, but the output is different.
The next question is that is there any hormone effect on singlet oxygen?
What actually there are two or three more other stories where we worked like that, and then what we actually thought is yes, indeed there should be a cross effect, crosstalk between these molecules. So if I have to put them in a series, I will put ROS at the top. So because ROS can generate JA, ROS can generate OPDA, ROS can generate SA. So all of these things we have shown in our studies. We found ROS is at the top.
Yeah. Thank you so much, sir, for your time and your patience in listening all of our questions. And thank you for your valuable time with us. I extend my sincere gratitude to you on behalf of my entire team of Biomics. I hope all participants are satisfied with the speaker's elaborate explanation and answering all your questions. Thank you so much for all. Once again, I thank everyone who joined with us. Especially, I thank our today's speaker for his excellent elaborations during question and answer session. Thank you so much, sir.
Thank you, and thank you, Partha. Thank you, whole team of the Biomics, and thanks everyone who were there, and who could bear me, because I know, I don't know, maybe sometimes maybe things were like slightly out of the way, because sometimes it's difficult, to really explain the things to a general audience. When you talk to people who are already working in that area, it's very easy. No, but sometimes it's difficult when you see some people are actually general audience. They do not actually have a they are not working on that particular area. So sometimes difficult. But I hope I tried my level, best to explain things in such a way that it could be understandable. And my apologies if I could not really make everyone understand the stuff. But I am open for the questions. Anyone can ask questions to me anytime. They can ping me. They can send emails or whatever they want to have. Even they want to work or collaborate. For students, the young students, they want to pursue their careers towards this research in this area. They are more than I'm more than happy to welcome all of them. And thank you. Thanks a lot.
Yeah, thank you so much, sir. Really, it was a very interesting session, and it were a great time having you. And it was a great experience in a research area of plant sciences. Thank you so much.
Thank you.
Yeah. Then a few announcements to the participants. Dear participants, our next session of today's talk falls at 6 p.m. as per Indian Standard Timing. The speaker of the second session would be Dr. Praveen Rahe, who is currently working as a scientist in National Center for Microbial Resources at National Center for Cell Sciences in Pune, at India. And he will be delivering his talk on biome, a reservoir for sustainable agriculture. So stay connected with the Biomics team. Stay connected for further updates. Join with us. Stay tuned. Thank you, everyone. Thank you so much. Thank you so much, sir. Thank you.
Thank you. Thank you. Thank you.
Thank you. Bye.